So as you know, I'm at the University of Michigan, and just by way of disclosure, since I'm going to be talking about Aducanumab, which is a drug owned by Biogen, I should mention that I do receive research funding not for the drug Aducanumab, but to study a different drug that's owned by Eisai, but kind of co-licensed with Biogen. I have not been involved in any of the Aducanumab studies. We have not participated at the University of Michigan. So the information I'm going to share is really what's out there, what's available, which is largely what's been presented at conferences by Biogen and is available also through their website. It's not yet published data, so it's not really out there in the general scientific literature yet. But as you may know, this
So what
drug,
I'm trying to do, Mike, is empty.
Oh, I'm getting some competing noise, so if everyone can mute. So this drug Aducanumab is right now, as we speak, being reviewed by the FDA as a potential treatment for Alzheimer's disease. And I know everyone is very interested in the answer to the question, will it be our next therapy?
Hey, how are you?
Since time is of the essence, I'm going to go ahead and just, for those of you who might have to leave early, I'm just going to flip to the answer to that question with my very next slide, which unfortunately is this, a big question mark. So what I'd like to do is spend the rest of the time sort of explaining why this is going to be, I think, a tough decision for the FDA and what kind of information they have to review in determining if this might be our next therapy for Alzheimer's disease. Just to start with, why is this such an important question? Well, as you probably know, it has been nearly 20 years since we've had the last approved therapy for Alzheimer's disease. And in essence, we have two types of medications that are available to slow down the progression of Alzheimer's dementia. There are the drugs that work on acetylcholine, and then there's the drug that works on glutamate. So both of them focus on some of the chemical changes that are happening in the brain when someone has Alzheimer's dementia. None of them are really directly addressing the underlying hallmark pathologies, the plaques or tangles of Alzheimer's disease. And as I mentioned, it's been nearly 20 years, back in 2003, since we last had an approved therapy. So there's a great demand for newer and better therapies. And a lot of research has focused on therapies that are more directly targeting the underlying pathology of Alzheimer's disease. So this is just a picture of some of the approaches that are targeting the amyloid, the main protein in the plaques that we see in Alzheimer's disease. So those amyloid plaques are composed of bits of the protein that sort of builds up from smaller fragments and accumulates or aggregates in the form of these plaques that can injure the nerves. And so one approach that has been widely studied in research is using antibodies that bind to the amyloid protein, either in its early pieces or in its more dense deposits. And by binding the antibody to the amyloid protein, then the body's own immune system can remove the sort of plaque antibody complex from the brain to see if removal of the amyloid leads to an improvement or a betterment of someone with the symptoms of Alzheimer's disease. So that's kind of the rationale behind the approach. But how do we get from an idea to an approved drug in the clinic? Well, this gets back to how drugs go through their stages of development. And typically here in the United States, as a drug is being studied, it progresses through sequential clinical trial phases. These are studies in human subjects. Even before drugs reach the clinical trial phase, there's a lot of preclinical development studies in the laboratory, in perhaps animal models of a given disease. But when drugs move into the clinical trial phases, they progress through phases 1 through 3, with the initial phase focusing on, is this drug safe? And so the very preliminary studies might simply be looking at what amount of this drug can be taken by an individual and over what period of time and what kind of side effects might they have. And sometimes these initial phase 1 trials are even not focused on individuals with the disease, but just a healthy volunteer to see where does the drug go in the body? Does it affect any particular type of body function? And again, at what dose and for how long can someone use the medication? If it looks safe at that stage, medications will move on to phase 2, where there's a larger group of individuals who are exposed to the medication. These are typically patients. There's often a placebo group to compare them to, and you're starting to look to see, is there a beneficial effect of the drug at the doses that we believe are safe, but we're still looking for any possible side effects that might develop when more individuals have the chance to take the medication and over a longer period of time. And so those studies may go on for several months. And then drugs can progress on to the kind of pivotal phase 3, which involve large number of individuals. And again, a placebo comparison where you're comparing the active drug to an inactive comparison. And you're really looking at what's the overall benefit of the medication versus the potential risks and how are those in balance. And so those studies can evolve, particularly in Alzheimer's disease, over years to try to track how people are changing or not as they're exposed to a medication. And typically for a drug to then become approved and available for clinical use, the FDA, when they review the information, they, at least in the past, were sort of historically looking for 2 well-done large studies, so 2 phase 3 type of studies showing evidence of benefit and acceptable risk. Technically, they could approve a drug on just a single pivotal study if it really had substantial evidence of benefit. No one knows exactly what that means. Some people have said, well, that means it has to be statistically impressive at not just the 5% chance level, but maybe at there's only a 1% chance that the findings could have happened randomly when you look at the data. So really you're looking at sort of the large pivotal trials to decide, is this drug something that looks promising? All right, so where does Aducanumab stand when we look at these sort of phases of drug development? So several years ago, we learned the results of one of the phase 1 trials of Aducanumab, and this one was focused specifically about, can this drug remove amyloid plaque from the brain? Since again, that's really the mechanism of action. Do we have proof that it can do that? So in this study, it was a fairly large study for a phase 1 study. There were some earlier smaller ones just looking at tolerability of the drug. But in this study, they looked at persons who either had mild cognitive impairment or mild dementia due to Alzheimer's disease. So all of these individuals had to have evidence of brain amyloid accumulation on PET scans looking for those amyloid deposits. So the left column here just shows representative baseline scans. When you do an amyloid PET scan, the tracer is binding if the amyloid is present, and the brighter kind of red signal indicates higher levels of binding. So at baseline, all of these individuals did have evidence of amyloid deposits consistent with Alzheimer's disease pathology. And individuals received treatment with either the study drug or placebo. These are infusions of the antibody. They were monthly infusions for a year, and you could receive either the placebo or one of 4 different doses. And they just looked to see what happens to the PET scan after a year of this type of therapy. And the right-hand column shows you the results, that there was a very clear dose-dependent removal of the brain amyloid, so that individuals receiving the highest dose, 10 milligrams per kilogram, essentially had amyloid PET scans at the end of the trial that would be considered normal. So they didn't really see any ongoing evidence of amyloid deposition, whereas those in the placebo group still were showing significant evidence of the amyloid deposits. So clearly a biological effect of the drug in what might be a beneficial way, but they also noted that as people were receiving the higher doses, they could also have some abnormal changes on other radiologic studies, with findings that have now been labeled ARIA, which stands for amyloid-related imaging abnormality. And these are changes that you would see on an MRI scan rather than a PET scan. And this just shows some examples of some of those abnormalities that can be associated with these antibody therapies. These pictures are not from the Aducanumab trials. They're from a trial of a similar agent called bapineuzumab. Essentially, there are 2 types of amyloid-related imaging abnormalities that you can get. You can either get swelling or edema, so that's called ARIA-E for edema. It shows up on an MRI scan as this area here, where there's kind of a brighter area reflecting some fluid accumulation in the brain and some associated swelling. This would be a fairly prominent area of ARIA-E. Sometimes it's much less prominent, just a little linear area here, or even a very, very subtle area here. These latter two are likely not to be associated with any symptoms, but this one here may very well be associated with symptoms ranging from things like headaches to nausea, or depending on where in the brain it occurs, here in the back of the brain, there might even be changes with visual, perceptual abilities, things like that. So that's one possible downside of these approaches, is developing this edema in the brain. The other is that the blood vessels, besides getting leaky in a way that can allow fluid to come in, they can also show evidence of sort of blood product leakage into the brain. So these little tiny black dots are tiny areas of blood products, or microhemorrhages. And so that's called ARIA-H for ARIA hemorrhages. And that also can be seen with some of these immunotherapies. So how often was this seen in the original phase 1b Aducanumab trial? Well, the results, the frequency of these radiologic findings depended on the dose, so much higher. This is specifically looking at the ARIA-E. So those with the highest dose exposure had a high percentage of ARIA-E, particularly if they were carriers of the ApoE E4 genotype of the apolipoprotein E gene. We know E4 has a higher association with Alzheimer's disease in general, but in particular may have a higher association with vascular deposits of the amyloid. So you may be at risk for some of these imaging abnormalities. So a relatively high risk in the E4 carriers at the high dose. And a sizable number of individuals did end up discontinuing the drug in the phase 1b trial, much less frequent if you weren't an E4 carrier or if you received a lower dose. So this was sort of a known consequence, and this started to inform future trials of the drug, because they also found that if you started the doses at a lower level and ramped up to the higher dose, you could avoid much of these side effects. Or if you paused dosing when any of these changes started to occur, often it resolved and people were able to resume dosing. So they did need to make some modifications as they planned future trials. But essentially, on the basis of the strength of this evidence, Aducanumab sort of skipped over phase 2 and went right into phase 3 studies to try to decide, does amyloid removal, does it help individuals with symptoms related to Alzheimer's disease? So 2 large-scale phase 3 studies were launched called eMERGE and ENGAGE. And these enrolled more than 3,000 individuals worldwide, individuals who either had mild cognitive impairment or early dementia due to Alzheimer's disease. So all had to have confirmed amyloid pathology. And in terms of their actual clinical characteristics, on scores like a mini mental state exam, a 30-point test of thinking abilities, scores had to range between 24 and 30. So again, very mild symptoms. On a severity of dementia rating scale, they had to be in the sort of equivocal, are they, slightly impaired, sort of pre-dementia, but they did have to have evidence of some degree of decline in their thinking on another neuropsychological measure. So this was typically picking up the memory loss that they were experiencing. And so individuals with that sort of defined level of disease were exposed to either the low dose or a high dose of aducanumab or placebo, with the goal of following them for 18 months to look at changes in the severity of their impairment on something called a clinical dementia rating scale. So the outcome that they chose, I'll talk about in a little bit, but just to show you what the individuals who enrolled in these studies look like, the 2 studies, again, eMERGE and ENGAGE, if you just kind of look back between the 2, left to right, they really recruited a very similar group of individuals. So average age, whether you were in one trial or the other, whether you received the placebo or the active drug, average age was around 70. There were about 50-50 men and women, generally a well-educated group with more than a high school level of education on average. Interestingly, about half or more were already on Alzheimer's disease medications, which is a little bit interesting since the majority of individuals were felt to have not dementia, but mild cognitive impairment, where there aren't any established medications. But about 80% of the group had mild cognitive impairment and about 20% had dementia due to Alzheimer's. And not surprisingly, there were a fair number of ApoE4 carriers in the group. And so again, the main thing that they looked at as they followed these individuals through the study was how they did on an assessment called the Clinical Dementia Rating Scale, which is a structured interview with the patient and with an informant, a family member or someone who knows them well, where you just kind of a conversation about how they're doing in terms of day-to-day things, talk about things that have happened in their life so you can test their memory for actual real-world events instead of just artificial things on pencil and paper. We test their ability to know date and time, things like that. And so it's a combination of testing and discussions from someone who knows them. And on the basis of that, the interviewer rates how they're doing across a number of areas. And their rating can be anywhere from they seem perfectly normal, so a 0 score, to they're showing a little bit of trouble, but really are still quite independent, which would rate a 0.5. And then higher numbers would signify higher levels of dependence or difficulty. And so you can calculate from this either a global clinical dementia rating scale, which is somewhat of an average, although it's not perfectly an average, it's weighted a little more in the memory domain, but you can sort of think of it as an average. So by and large, people entering the trial had a global score of 0.5. So most of their individual ratings fell in this 0.5 box. So not normal, not really in the dementia range, but kind of in this mild cognitive impairment range. And so they wanted to track and see how these ratings changed over time as they progressed through the trial. Well, they also built into the trial what was called a futility analysis, which said that we're going to look at the data when roughly half of the individuals have made it through the trial to see if at that point the results look promising enough to continue with the trial. And they had kind of a built-in rule: if at that point it looked so unlikely that the results would ultimately be positive, they would stop the trial. And that's in fact what happened. So in March 2019, Biogen halted these 2 large trials of aducanumab because their preliminary look at the data suggested that they weren't going to see a beneficial outcome. And so this was a really shocking event when it occurred. And unfortunately, many of the trial participants found out about it only as it hit the news waves instead of through their sites where they were participating in the trial, because it got publicized right away that the trial was coming to an end. And the question is, why did the trial get stopped? Well, at the point that they stopped the trial, they'd actually frozen the data through late 2018, but they announced the halting of the trial in March 2019. So once they halted the trial, they still had a few more months of data that they could look at. And when they looked at all of the data that was available up until the time that the drug was halted, what they basically saw was that one study was showing a potential benefit and the other one was not. So if you look at that key outcome measure, that clinical dementia rating scale, higher numbers on that mean more impairment. So the placebo group was gaining numbers on that clinical dementia rating scale at a faster rate than the highest treatment group. So there was about a 22% difference in the amount of decline between the placebo group and the highest dosing group of the aducanumab in the EMERGE trial. But in the ENGAGE trial, essentially there was no detectable difference. If anything, the treatment group had a slightly worse score. The highest treatment group had a slightly worse score than the placebo, but for all intents and purposes, really no discernible evidence of a difference in the ENGAGE trial compared to, at least at the final data point available, what looked like a slowing of decline. Again, this was just one of the measures they looked at. If we focus on the EMERGE trial, what about the other measures they looked at? Well, in a similar trend, all of the other kind of key outcome measures, whether it was a cognitive test called the MMSE, where higher scores are better, the highest treatment group ended up with a statistically higher score at the end of the treatment phase. On this particular test, a lower score is better. And again, the highest dose treatment group ended up with statistically better score. And if we look at activities of daily living, higher scores are better on this, and the highest treatment group ended up with a higher score at the end of the study. Now, these graphs show the change from baseline. They aren't the actual MMSE scores. They're just telling you how much the MMSE scores on average changed over time. So they're really, in a sense, kind of magnifying the difference. If you look at the absolute values, like for example, on the mini mental state, on average, people were starting off with an MMSE score around 26 and change. And so in the placebo group, they dropped to a score of about 23 by the end of the trial. the highest dose treatment group was better off by 0.6 points. So they were at about 23.6. So on a percentage basis, that was 18%, but in absolute, it was only half a point. And so you could argue, could I tell someone, could I tell that they were benefiting if they're a half a point different on an MMSE score? I think that would be a difficult judgment to say that I could tell they were clearly different. And same thing for some of these other measures. They statistically ended up being significant, but the question is, clinically, can we tell folks apart with this amount of difference at the very last time point in the 18-month trial? And again, this was for the eMERGE trial. For the ENGAGE trial, if you look at all of those other outcomes, there was really no difference between the low dose, high dose and the placebo group. So in a sense, one positive trial and one negative trial. So what does all this mean? Well, Biogen really tried to look and see, is there something that would explain why these two studies produce such different results? And one interesting feature that happened during the course of these studies before they were halted was that there was a protocol amendment that allowed individuals who were E4 carriers to go to a higher dose of the drug than had originally been intended. Initially, if you were an ApoE4 carrier and were randomized to the high dose group, you only went up to 6 milligrams per kilogram, in part because of that concern about the ARIA side effects. But as we gain more information in general in the field about how to manage ARIA or prevent ARIA, they then allowed individuals to go all the way up to the maximum dose. But that didn't happen at the start of the trial. That happened as the trial was ongoing. And it turns out that the two trials, even though they were running simultaneously, more individuals in the eMERGE trial who were E4 carriers had the opportunity to get to the higher dose compared to individuals in the ENGAGE trial. Did that make the difference between the two study outcomes? And I think that's a huge unanswered question, but when you go back and look at the data and say, let's just focus on the folks who did receive the highest dose, which again was only a subset of individuals because many individuals were randomized to the lower doses, or in some cases, they never made it up to the higher dose before the trials were halted. But if you just focus on those who received at least 10 uninterrupted doses at the highest 10 milligrams per kilogram, what would you see then? Well, if you look at the PET scans, not surprisingly, compared to the placebo group, if you look at the amount of amyloid that's basically staying the same if you were in the placebo group, those receiving the highest dose of aducanumab clearly had a drop in their amyloid brain levels. But if you look at that primary outcome measure in the eMERGE, as we saw previously, it looks like there was a difference between the placebo group and the treatment group. And it looks like you're starting to see a difference now, perhaps with the ENGAGE group too. Now, you really can't do any statistical analysis of this because this wasn't a designed trial of the high dose alone. This was what's called a post hoc analysis kind of after the fact. So it's really hard to know what to make of that, but just based on the proposal that maybe the drug would have been effective if everyone had been randomized to just the highest dose, the 10 milligrams per kilogram versus placebo, Biogen looked at that and applied to the FDA and said, can we apply for approval based on this sort of reanalysis of our trial data? And the FDA said, yes, you can certainly submit an application. And they also granted Biogen permission to relaunch the trials, so to bring patients back and to allow them to re-enter the trials. But at this point, everyone who's re-entered is getting the active drug. There's no longer a placebo group. And so essentially all the data from here on out is going to be just of the active drug trial, not for no longer any type of placebo comparison. But as of August, Biogen did collect all the data, submit it to the FDA for its review. Now I focused so far on just the potential clinical benefits of aducanumab. What about those downsides, that ARIA? How often did that happen? And if you focus again on just the folks who received the highest dose, the 10 milligrams per kilogram, you can clearly see that those getting the active drug had a much higher likelihood of the edema, so 35% versus only 3% in the placebo group. Also a much higher chance of the microhemorrhages, 19% versus 6%. Fortunately, these were not often clinically symptomatic, but people did in some cases experience headache or dizziness or visual disturbances. Generally, the edema resolved over a period of weeks, and many individuals were able to, if they needed to pause their treatment temporarily, they were able to resume it later on and continue on. There's actually a very interesting and dramatic example of edema in an individual, not in the placebo-controlled study, but later when they went into the open label, when they were allowed to re-enroll. One particular participant developed a very dramatic case of the ARIA edema and had to have a lot of intensive therapies. And interestingly, he is a retired neurologist, and so he actually is an author on the case report describing his experience with this particular compound. But in any event, we now have, both some evidence of potential benefit, but also some potential side effects that need to be taken into account. So at this point, the FDA convened an external advisory committee. So these are not individuals who are going to make the ultimate decision about whether to approve this drug or not, but these are sort of experts in the neurologic field. And they were asked to review the data and answer several questions, two of which are listed here. And they were basically saying, how much do you think the data shows evidence of a benefit? And they were asked to focus specifically on eMERGE, the one that, again, looked like it was showing promising results at the time that the studies were halted. If you just focus on it, put aside the other one that didn't clearly show a benefit, is that enough to show strong evidence? Could you approve this drug on the strength of this one study? And the advisors pretty overwhelmingly said no. And they said, well, what if we take everything together, including kind of reanalyzing the data, maybe thinking about how this protocol amendment and the different dosing regimens might have impacted things, along with the evidence that, from the earlier phase 1 trial that we can clearly remove amyloid from the brain with this drug. Is that enough to say that's primary evidence of the effectiveness? And again, a pretty overwhelming no vote. Now, this doesn't mean that the FDA won't approve aducanumab. They are not bound to follow the advice of their advisory committee, but obviously it's something that needs to be taken into account. In a sense, it was sort of asking the advisory committee, could we consider that one trial strong enough to use that as the pivotal trial alone for approval? And the response was generally no. And in part, it was asking them to sort of set aside this other equally well done trial that didn't clearly show a benefit. And so the FDA has essentially said at this point, we need more information. So they went back to ask Biogen for more data. We don't know what that request was for, but when Biogen submitted it, the FDA said, this is enough new information that we need to take more time to consider it. So we're delaying our decision deadline from what would have been March to now early June. And so we don't know what that extra data request consisted of. We do know that the statisticians at the FDA had some questions about whether even the eMERGE trial was truly positive, or was this difference between the placebo group and the high dose treatment group, was it really driven by the placebo group sort of suddenly worsening in an uncharacteristic way, rather than the active drug suddenly having a benefit by the final visit? Or was it the case that when individuals develop some of these side effects like the ARIA, if you developed ARIA, you were told you couldn't have the drug for a while, there might have been temporary hold on the drug, so you sort of knew that you were likely getting the active treatment, and so that might have led to some unblinding where people knew which therapy they were getting, and perhaps that affected their behavior or the ratings in the trial. So there are a lot of questions still, and how do you really reconcile two well-done studies that unfortunately were interrupted before their desired endpoint and ended up with relatively conflicting results? So that's kind of why I left this with, I don't know how the FDA is going to rule. There's more data than what I've shared that maybe that they're looking at. I think there's some interesting data from some of the biomarkers that were looked at during the study, but this is really limited data. Like there were some individuals who had tau scans, but of the 3,000 or so participants, it was only about 30 who had tau scans. Biogen has shared, quote, representative findings. So tau is looking at the deposition related to the tangles of Alzheimer's. So again, the brighter signals show more evidence of tau deposition. So here in the placebo group, you can see the tau deposits down deep in the kind of temporal lobes look pretty similar or increasing from beginning to the end of the study, whereas in the high dose treatment group, you could argue that seeing the amount of red here is lessening over time. So, this would be a potential promising biomarker, but whether that actually correlates with a clinical benefit, I think is really unknown at this point. So that's why I end up with the big question mark. And it's not just will aducanumab be approved, but it's also, if it is approved, in whom, because there were very specific rules about who could be eligible in the trial. It's not things that we just look at in the course of a clinical visit. So we might do a mini mental state exam during a clinical visit, but not some of these other measures that would let us know whether someone is truly eligible. And in particular, amyloid PET scans are not reimbursed by insurance companies right now. So there would have to be some way of knowing who has brain amyloid, whether that would change PET scan coverage or whether lumbar punctures would need to be done. And still some unanswered questions. If this is ultimately approved, how long should we give it? Is a year and a half, 78 weeks the right amount? Or do people stay on it indefinitely? Do you stop? Do you repeat a PET scan, see if the amyloid's gone then? So unfortunately, a lot of yet to be answered questions. So we're all gonna have to just wait, I think, and see what comes out of the FDA review. I think that's my last slide. I know I'm at our time, but maybe a quick question or two I could entertain.
I think one question which would be really helpful is kind of what's the takeaway for the average lay person here when they hear in the press, oh, there's potentially a new drug. What is kind of your one sentence takeaway that you hope everyone understands from your presentation?
Yeah, I mean, I think there are probably two things. One is this is not a drug for everybody with Alzheimer's dementia. It's really people with very early clinical symptoms. Most of them, again, were at the pre-dementia stage. And even in that group, the drug looks very powerful in being able to remove amyloid. The question is, how helpful is that clinically and at what risk? And so that's the dilemma right now.
Thank you very much. And we have one other question. What is the full name of the assessment? ADCS-ADL MCI.
Oh, that's the Alzheimer's Disease Cooperative Group Activities of Daily Living. So it's kind of a survey of how someone does day-to-day tasks, but it's focused specifically for people with milder impairment. So that's what the mild cognitive impairment stands for. So it's not going to ask things like, do you need help using the bathroom? It's going to be more like doing your finances, transportation, shopping, meal preparation, and just kind of surveys different areas and says, do you do this on your own? Do you need some prompting? Does someone need to assist? And so it's kind of a rating of independence with daily tasks geared towards individuals with very early symptoms.
Okay, well, those are all the questions that came up in the chat. I'm sure people have other thoughts, but I know that you have another time commitment as well. And yes. I want to thank you so much for fitting us into your schedule. This was really, really informative. And thank you so much for being here this morning.
Great. Thank you so much. All right. Take care.
Have a great day.